Hi, welcome to our channel of IGNOU Audiobooks, Indira Gandhi National Open University School of Law, SOL. Postgraduate Diploma in Intellectual Property Rights, PGDIPR. MIP106 Plant Varieties Protection, Biotechnology and Traditional Knowledge. Block 1 International Conventions for the Protection of New Plant Varieties. Unit 1. Objectives of Plant Varieties Protection and Plant Breeders' Rights, PBRs. 1.1 Introduction. The evolution of the intellectual property rights regimes has always been influenced by the prevailing political and economic situations. In addition, the area domain of the innovations that are intended to be protected has led to emergence of a family of IPR systems that are applicable for different types of intellectual creation. Patent laws have traditionally exempted plants from the scope of protection. In this context, the Plant Variety Protection (PVP) a form of IPRs for plant varieties is a relatively recent phenomenon in the long history of IPRs. Till the early 1990s, PVP remained almost exclusively a feature of developed countries. The major watershed period related to IPR protection for plant varieties resulted from the Uruguay Round of General Agreement on Tariffs and Trade, GATT, concluded on 15 April 1994 and known as the Agreement on Trade-Related Aspects of Intellectual Property Rights, TRIPS, Agreement. During the negotiations, the developed countries and transnational corporations lobbied for expansion and introduction of uniform IPRs regimes for protection of plant varieties globally. The implications of the extending regimes of IPRs, particularly in the developing countries, witnessed heated public debate at national and global forums that cut across wide range of sectors such as food and nutritional security, biological diversity conservation and environment degradation, economic, social and cultural development and ethics and human rights. However, as acceptance of the TRIPS agreement was one of the conditions of entry in the World Trade Organization, WTO, many countries opted for the obligations of TRIPS to harness the benefits of WTO. The obligations included that the countries provide an effective system of plant variety protection within a specified time frame. This has significantly accelerated the spread of PVP systems across countries. The form of intellectual property for protection of plant varieties is known widely as plants breeders' rights, PBRs. The PBR, like any other IPR system, allows the plant variety owner to prohibit specific unauthorized uses of the variety. The plant breeders' rights, PBRs, which are more comprehensive in the coverage of varieties to be granted legal protection, have a recent origin, even in the developed world. While there are universally accepted minimum standards for other forms of IPRs, most of the countries have opted for sui generis legislations that offer less stringent protection than patents. PBR systems, like other IP systems, has three major components. 1. Definition identification or protectable subject matter. 2. Requirements that must be met to receive protection. 3. Rights of the variety owner. Before we try to understand the details of legislations for obtaining PBR in subsequent units, in this unit the focus would be to understand the subject matter, including what are plant varieties, basic principles involved in plant breeding and the need for separate PBR system. 1.2 Objectives. After completing the study of this unit, you should be able to times acquire a basic knowledge of taxonomy of plant variety. Times explain some of the key features of plant breeding. Times describe the process involved in developing a new variety. Times explain the benefits of a PBR system. 1.3 Plant variety. It is well known that plants are a major source for human beings for their needs, including food, fodder, fiber, fuel, shelter, and products of industrial and pharmaceutical importance. In addition, the plants make tremendous indirect contribution in maintaining greater resilience in the ecosystems and also in providing for recreational, inspirational, and spiritual comforts to human beings. 1.3.1 Plant classification. The plant kingdom is vast, and throughout ages human beings have been interested in the study of plants. Various explorers discovered new lands and came across new plants of interest. Therefore, the need arose to describe, classify, identify, and names plants. Carolus Linnaeus, a Swedish physician and botanist, published in the mid-1700s a two-volume work Species Plantarum. Linnaeus grouped the species according to their reproductive parts and classified them into many divisions and subdivisions. Family is a division within the plant kingdom, usually where the classification process takes on significance. As at this level, the similarity between plants is often easily recognizable by the layman. The family is subdivided into genera, which are further subdivided into species. An example would be of family Solanaceae, the potato or nightshade family, which includes some 100 genera and nearly 2,500 species and includes major crop plants such as potatoes, tomatoes, bell peppers, eggplants, tobacco, and the garden petunia. Although the rank of species is an important botanical classification, it is clear that the plants within a species can be very different. Farmers and growers need plants that are adapted to the growing conditions in their local environment and which are suited to the cultivation practices employed. Therefore, farmers and growers use a more precisely defined group of plants selected from within a species called a plant variety. A case in point could be that in India, there are nearly 1,000 mango varieties which have specific ecogeographical requirements for optimum growth and fruiting. Some of the well-known commercial varieties include Dasheri, Langra, Chausa, Bombay Green and Fajri in North India, Banganapalli, Totapuri, Neelam, Peri, Suvarnarekha, Malgoa, Kalapadi and Rumani in South India, Alfonso, Kesar, Mankurad, Fernandin and Vanraj in Western India and Langra, Fajri, Chausa, Jardalu, Himsagar and Malda in Eastern India. 1.3.2 Naming of plants. Further, it is important that the names of the plants are accurate and unambiguous. While most of us are much more comfortable referring to the plants by their common names, the problem is that these names are generally specific to a geographical region and therefore not universally accurate. For example, eggplant, aubergine, melongene, guinea squash, berinjena, garden egg, apple, patlican, melanzane are some of the names by which the plant we call as brinjal is referred to. In contrast, the same plant has one scientific or botanical tilde name, Solnum melongena, acknowledged worldwide. The major advantage of scientific naming of plants is that there is only one name per species which is recognized and documented worldwide and is same in any language. There are three basic rules pertaining to the scientific name. 1. It always has no parts, the genus, capitalized first letter, and species, lowercase. Second, the order is genus first, species second, like Gupta Anil. And third, both. Names are always set apart in the text by underlining, italicizing, or bold-facing. The application of scientific names to plants is governed International Code of Botanical Nomenclature, ICBN, which provides a single set of rules accepted and followed throughout the world and periodically revised and updated. 1.4 Origin of plant breeding and crop improvement underscore. In addition to the enormous genetic diversity between plant species resulting in the large number of plants we see, there also exists large genetic variability within a species. This genetic diversity is essential for any species to maintain its reproductive vitality, resistance to disease, and ability to adapt to changing conditions. In the absence of this genetic underscore diversity, it would not have been possible to identify plants that exhibit variation for the traits or plants of interest. It, therefore, formed the basis for genetic improvement both at the plant level, through plant selection, and at the varietal level through varietal choice. The early farmers recognized that certain species were more suitable than others for their needs and that within populations, some plants were more suited to their specific requirements. The efforts resulted in developing combination of populations and growing environments that result in phenotypes that provided the optimal net benefit to the farmer. Underscore. As the improved crops were further carried and introduced to new locations, the natural intercrosses with new wild populations provided opportunities for increase in genetic variation through the interaction of the environment with the genotype, the genetic makeup, and the resultant expression of the genotypes in the form of the phenotypes, morphological appearance. Darwin had observed that in any population of plants or animals, individual members carried hereditary traits which are slightly different from one another. It is these traits which could give the member a better chance of adaptation. According to the theory proposed, these better adapted individuals would get more food, be healthier, live longer, and, most importantly, have more mates in staying alive long enough to successfully reproduce and pass on their traits more frequently to the next generation. Further, with successive generations, traits become more obvious in the populations and possibly after thousands of generations form a new species. The earlier crop improvement efforts were aimed at selecting the naturally occurring variants. Systematic plant breeding began by the end of the 18th century, when the innovative farmers realized that considerable further progress was possible by systematic selection. 1.4.1 Beginnings of systematic plant breeding. The early plant breeders made conscious attempts to predict the performance of plants that could be expected from one generation to the next. Each year, the best appearing individuals of a generation of plants in the field were selected, and seeds for next sowing were collected from these superior individuals. Only practicing this selection for each subsequent generation proportionately resulted in more superior individuals, and after many generations of seed selection, a superior new variety of a plant species could be bred from the original inferior variety. Initial efforts to breed plants were somewhat of a hit or miss process, as they were carried out without a clear understanding of the genetic mechanism involved in inheritance. The refinement in the selection process came when the importance of ancestry was realized. Mendel in 1866 worked with common pea plants, Pisum sativumi, and selected 7 traits that could be easily recognized and apparently occurred only in one of two forms, such as flower color, red versus white, plant height, tall versus dwarf, seed coat, smooth-coated seeds versus wrinkled seeds, pod length, long pods versus short pods, and so on. He observed that these traits show up in offspring without any blending of parent characteristics. Through the selective crossbreeding for over many generations, he proposed the laws of heredity that explained the expression of the characteristics and their inheritance by later generations of plants. 1.4.2 Genotype and phenotype. At the time that Mendel did his research, there was no knowledge of chromosomes, cell structure, fertilization, mitosis, and meiosis, which is now common knowledge. Geneticists distinguish between genes and their expression as genotype and phenotype. A genotype is the actual set of genes or genetic makeup of an organism, whereas a phenotype is a measurable characteristic of an organism. Mendel's findings, therefore, show a remarkable use of observation and deduction which was quite ahead of his time and linked the genotype of the pea plants with resultant expression of the genetic structures as observed in its phenotype. 1.4.3 Mendelian genetics. Since pea plants are capable of self-fertilization, Mendel was able to produce true breeding plants, that is, plants of the second generation had consistent traits with those of the first generation. A true breeding tall plant, for example, would only produce tall offspring. He performed monohybrid crosses, meaning that the experiment was carried out between two strains of plants that differed only in one characteristic. For each of the 7 phenotypes, they crossed parents of different phenotypes to see what resulted. He referred to the two parental plants as the parental generation, P generation, and the resulting offspring were called the first filial or F1 generation. He noticed that by crossing pure tall plants with pure short plants, all the new pea plants in the F1 generation were tall, the one trait not expressed in the offspring, in this case short plant, he called a recessive trait. The following generation, F2, consistently has a 3:1 ratio of tall to short plants. He hypothesized that there is a factor that makes pea plants tall and another factor that makes pea plants short, and when the factors are mixed, the tall factor seems to dominate the short factor. The individual inherits one such unit from each parent that are passed on to descendants unchanged, and a trait may not show up in an individual but can still be passed on to the next generation. It was not until the early 20th century that the importance of Mendel's ideas was realized. In modern-day genetics, we use allele forms of expression or gene instead of what Mendel called factors. An allele that can be suppressed during a generation is called a recessive allele, while one that is consistently expressed is a dominant allele. It is also now established that in diploid plants, there are 2 sets of chromosomes, and for each gene, there is a pair of alleles, one in each chromosome. The plants or individuals who have 2 copies of the same gene, e.g., round or wrinkled are described as homozygous, and individuals which received a different type of gene from each parent, round or wrinkled round, are described as heterozygous. In this case, round being the dominant trait, the phenotype of the plant would be round, though the genotype round wrinkled. It is important to realize that, in this experiment, the starting parent plants were homozygous, that is to say, they each had 2 identical or alleles of the gene For the traits, the plants in the FL generation were all heterozygous. Further, after performing dihybrid crosses between plants that differed in two traits, such as seed color and pod color, Mendel formulated the principle of independent assortment exercise based on the expression of the characteristics and their inheritance by later generations of plants. Let us understand their applicability in plant breeding and variety development. As a first step of selection process, let us identify heterozygous plants for two traits, namely height and flower color. For considering a dihybrid cross, let us designate capital T for the dominant tall allele and the lowercase t for the recessive short allele and capital C for the dominant colored flowers allele and the lowercase c for the recessive white flowers allele. As allele for tall plants and colored flowers are dominant, a tall plant would have the genetic expression TT or TT, while plants with colored flowers would have the genetic expression CC or CC. The phenotype of the heterozygous plant will be tall with colored flower, as these are dominant traits, and the genotype will be TTCC. Since the gametes are TC, 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 TC and this is a self-fertilization, the gamete types shall be the same for both parents. Let's see below the Punnett square, a diagram named after Reginald C. Punnett, used to predict an outcome of a particular cross or breeding experiment and represents visual representation of Mendelian inheritance. Male gametes. Look at the screens. Now let us count the number of plants with different phenotypes. You will find these are tall and colored flower is equal to 9, tall and white flower is equal to 3, short and colored flower is equal to 3, and short and white flowers is equal to 1. Therefore, it can be concluded that different pairs of alleles are passed to offspring independently of each other, and the same ratio of 9, 3, 3, 1 appeared among the offspring. Therefore, for example, a pea plant's inheritance of the ability to produce tall plants instead of short ones does not make it more likely that it will also inherit the ability to produce colored flowers seeds in contrast to white ones. 1.5 Plant breeding and variety development. Plant breeders have played a pivotal role in improving the crop's productivity, particularly as witnessed in the second half of the last century. The self-sufficiency in food grains achieved during the Green Revolution would not have been possible without the huge genetic improvements made to the crops. Recent scientific and technological developments have allowed a greater rate of improvement. Today, breeding has evolved as a sophisticated, high-investment business, and the breeding programs plan and look years ahead to the needs of farmers, consumers, and the environment. Plant breeding can directly improve the performance of crops in different ways. Some of the broader objectives are given below. 1.5.1 Increasing the productive yield. Developing crop varieties which convert more of their biomass into productive yield is the single biggest contributor to improved crop output. 1.5.2 Physical characteristics. Changing a crop's physical structure can also contribute to increased yields. Among the most spectacular development in crop improvement was transfer of dwarfing gene from Japanese wheat cultivar, Norin 10, which began a new era in crop improvement with the development of dwarf varieties. 1.5.3 Pest and disease resistance. There are numerous examples of devastating plant disease impacts such as the epidemic used by grassy stunt virus which destroyed more than 160,000 ha of rice in Asia, or the famous Irish potato famine in 1845 that lasted for 6 years and killed over 1 million men, women and children in Ireland and caused another million to flee the country. Therefore, selection and development of plant varieties that are resistant to pests and disease has been a major area of concern for plant breeders. While plant diseases can be partially controlled by use of agrochemicals and by proper cultivation practices, but incorporating inherent disease resistance in plant varieties remains the first choice for disease control. 1.5.4 Change in maturity duration. Plant breeding technology has brought major improvements in the uniformity with which crops ripen and are ready for harvest. While short-duration crops reduce cost of crop production, uniform maturity makes improved ability for mechanized harvesting operations. 1.5.5 Desirable agronomic factors. Improving crops' ability to cope with a range of other agronomy factors is another important objective. For example, for fodder crops, tallness and profuse branching are desirable traits for breeding new varieties. 1.5.6 Qualify improvement. Developments in plant breeding have brought significant gains in crop quality. This aspect is particularly important in horticultural crops. Many of horticultural crop cultivars, varieties with unique qualities are admired and appreciated so much so that they become the basis for entire industries. Examples include the Bartlett Williams pear, which dominates fresh fruit production and is almost the only cultivar used for processing, Hayward kiwi fruit, Karmari pistachio, smooth cayenne pineapple for processing, clementine, mandarin, and dwarf Cavendish banana.
1.5.7 Wider adaptation. The modern-day intensive cropping systems require varieties that can be grown in all the seasons as well as in all the environments. Failure to maintain vigorous growth during periods of climate extremes limits the usefulness of many crops. Varieties with traits of wider adaptability are desirable as the same crop variety can be grown in different conditions. Also, one of the major challenges for the plant breeders today is incorporating traits in crops to adapt to significant impacts of climate change, while at the same time providing food for a growing P9-ulation. 1.5.8 Improved nutritional aspect. At a fundamental level, food is viewed as a source of nutrition to meet daily requirements at a minimum in order to survive. There is now considerable evidence for substantial genetic variation within species for many of the health ameliorating properties. Consequently, plant breeders today are experimenting with opportunities to maximize the health ameliorating properties of different species through plant breeding. The aim is to develop functional foods which are modified food or food ingredient that may provide a health benefit beyond the traditional nutrients it contains, and developing plants with nutraceuticals or substances that may be considered a food or part of a food and provide health benefits, including the prevention and treatment of disease. 1.6 Approaches for development of new varieties. Mendel's discovery was the beginning of modern science of genetics. The example given, though may seem complicated, is among the simplest from plant breeding point of view, as it only covers two characteristics, height and flower color, controlled by two independent genes. As the science of plant breeding progressed, many exceptions to these rules were discovered. Many useful characteristics, such as yield and quality, for example, were found to be controlled by the interactio tilde of very large numbers of genes about inheritance, of which was not much known. Thus, the development of new variety is a time-consuming, complex, costly, and skilled operation. Let us examine the general techniques to produce plant varieties. 1.6.1 Conventional breeding. The finer techniques vary between crop species, but the scientific principles of plant breeding remain unchanged from Mendel's first discovery. Conventional plant breeding involves crossing carefully chosen parent plants, then selecting the best plants from the resulting offspring to be grown on for further selection. For example, in crop cereals, hundreds of individual crosses are carried out by hand to create seed for the F1 generation. The resulting F1 plants are uniform. But in the following generation, several 100,000 different plants are produced. Because of the way genes works, the new combinations produced from each cross are not revealed until the second, F2, generation. It is this enormous diversity of new gene combinations which may hold the key T6 a successful new variety. The plant breeder's task is to select the plants most likely to meet his breeding objectives. Seed from the best of these F2 plants are grown on in small rows or plots, and the best plants again selected. This process is repeated year after year until only the very best plants remain. As promising new lines emerge, tests are conducted on each plot to assess factors such as yield, disease resistance, and quality. Once the best lines are purified to ensure that every plant has the same characteristics, the process of multiplying seed begins. Since each plant contains many thousands of genes, and the breeder is seeking to combine a range of traits in one plant, such as high yield, quality, and resistance to disease, developing a successful variety is an extremely lengthy process, up to 12 years. In the case of cereals, even longer for potatoes and horticultural crops. 1.6.2 Hybrid breeding. Hybrid seeds are an improvement over open-pollinated seeds in terms of qualities. Hybrid seeds are developed by the hybridization or crossing of parent lines that are pure lines produced through inbreeding. A critical requirement of hybrid production is that the parents be unidentical. It is this divergence of genotypes of the parents that expresses in hybrid vigor, or heterosis in the F1 hybrid seed. The emphasis for development of hybrid varieties has been in cross-pollinated crops, such as maj, primarily because its male flowers tilde, tassels, and female flowers, incipient ears, are separate and easy to handle, thus proving economical for the production of hybrid seed. However, commercial hybrids are now available for many crops, including self-pollinating species. 1.6.3 Modern techniques for crop improvement. With increasing knowledge and improved technology, breeders have developed ways to enhance the speed, accuracy, and scope of the breeding process. There are several ways to reduce the lengthy interval between the first cross of selected parents and establishing true breeding lines of promising new varieties. For example, maintaining parallel selection programs in two different environments allows two generations to be produced each year. Latest developments in genetic science have greatly improved our understanding of how plants behave, offering additional ways to enhance the breeding process. Interspecific and intergeneric hybrids produced from a cross of related species or genera often do not normally sexually reproduce with each other and plant breeders make use of tissue culture techniques to produce progeny from such crossings. For example, the cereal triticale is a wheat and rye hybrid. Failure to produce a hybrid may be due to pre- or post-fertilization incompatibility in Such case is the embryo resulting from an interspecific or intergeneric cross can sometimes be rescued and cultured to produce a whole plant, and the technique known as embryo rescue. Mutation breeding makes use of mutagenic agents, such as radiation-arid certain chemicals, to induce mutations and generate genetic variations from which mutants with desirable traits to be bred with other cultivars may be selected. Single seed descent produces very small plants under restricted growth conditions. Large numbers of these plants are cultivated under controlled condition, with 2 or more generations produced in a year. In potatoes, mini-tuber breeding speeds up the slow multiplication process by producing miniature plants under greenhouse conditions. 1.6.4 Biotechnology and plant breeding. Revolution in plant breeding was witnessed with the discovery of new recombinant DNA techniques that allowed specific identification, isolation, and alteration of genes and their reintroduction into living organisms. Direct manipulation of DNA, or genetic engineering as it is called, is now regularly being used to complement classical plant breeding to achieve useful practical outcomes. In plant breeding, genetic engineering and other molecular biology tools allowed the creation of several new methods such as DNA fingerprinting, DNA markers that accelerate the relatively slow process of classical breeding. Marker-assisted breeding is increasingly being used by the breeders to determine whether desired traits are present in a new variety at an early stage in the breeding program. The science of genomics allows scientists to map genetic makeup, or genome, of crop species, and identify the exact position and function of individual genes. Genome mapping has revealed striking similarities in the genomes of different crop species, such as rice, wheat, barley, and rye. This information is already helping to broaden the scope and precision of current breeding programs. The scope of plant breeding continues to expand in the 21st century. Genomics, marker-assisted breeding, and RNA interference, RNAi, cRNA. Cisgenics are increasingly effective in accelerating commercial breeding, identifying the functions of physiologically relevant ends, and in allowing crop traits to be modified. 1.6.5 Genetically modified plant varieties. Genetic engineering techniques allows plant breeders to bring together in one plant useful genes from a wide range of living sources, not just from within the crop species or from closely related plants. In most cases, the aim is to introduce a new trait to the plant which does not occur naturally in this species and therefore cannot be incorporated in the variety through conventional breeding methods. It allows for the transfer of only one or a few desirable genes, thereby permitting breeders to develop crops with specific beneficial traits and reduced undesirable traits. The transgenic varieties have genes inserted into them that are derived from another species which can come even from across kingdoms. Some transgenic plant varieties, such as those with resistance to the herbicide Roundup, those producing protein Bt, have great potential to reduce the amount of land needed for farming and also to reduce the use of chemical pesticides while reducing the incidence of pesticide resistance. Other technical strategies involving transgenic varieties being pursued include using genetically modified plants as the production platform to manufacture industrial enzymes for use in pre-processing feedstock for cellulosic fuel manufacture. 1.7 Commercial plant breeding. Plant breeding is one of the first things in commercial seed production which entails a series of quite distinct operations and responsibilities. These include source seed production, seed multiplication, quality control, compliance with regulatory requirements, and marketing. Large, Modern commercial seed companies take responsibility for all of these operations in an integrated firm. 1.7.1 Maintaining genetic diversity. Genetic diversity is essential for any species to maintain its reproductive vitality, resistance to diseases, and ability to adapt to changing conditions. Since the dawn of civilization, economically useful plants have evolved thousands of locally adapted genotypes because of natural and human selection. These wild species, landraces, or folk varieties have the genetic reservoir of useful genes adapted to low input conditions and conferring resistance to various biotic and abiotic factors. One of the most important components of plant breeding program is maintaining as extensive as possible gene pool from which new traits can be selected. In the absence of this genetic diversity, it will not be possible to identify plants that exhibit variation for the traits or plants of interest. Thus, the full spectrum of plant genetic resources of any crop consists of diverse type of collections such as those derived from centers of diversity, those derived from centers of cultivation, and those derived from breeding programs. Some of the valuable genes found in plant genetic resources have provided tremendous boost to the crop improvement programs. It is important to mention that the gene banks established across the globe conserve the valuable genetic diversity within past and present varieties, as well as landraces and wild relatives of cultivated crop species. 1.7.2 Notification of variety. The developments in the seed industry in India, particularly in the last 30 years, are very significant. Introduction of New Seed Development Policy, 1988. 1989, transformed the very character of the seed industry and stimulated appreciable investments by private individuals, Indian corporate and MNCs in the Indian seed sector. Before commercialization of any new crop variety, each variety has to pass through a phase of evaluation. The breeders contribute their best entries on the basis of evaluation carried out in their local programs for testing in the initial yield evaluation trial, let entries qualifying from yield, disease and quality point of view in IGNOU are tested in the Advanced Varietal Trials, AVT, or Coordinated Varietal Trials, CVT. Successful varieties are identified for release and notification. The official trials are conducted, in most cases for a minimum of 3 years, to test each new variety for many characteristics which together determine its uniqueness, its genetic uniformity, and its value for cultivation and use, VCU. The proposal is then submitted for approval of the Central Subcommittee on Crop Standards, Notification and Release of Varieties. 1.7.3 Genetically modified varieties. In addition to the tests described above, varieties produced using genetic modification must also pass through a separate process of regulatory scrutiny. The regulatory clearances of GAC under Ministry of Environment and Forestry, Government of India is mandatory for conducting any genetically modified crops field trials in India, which specifies that no GM crops can be marketed until they have been assessed and approved in terms of human health, food safety and the environment. The cross-border movement of genetically modified organisms, GMOs, is regulated at a global level under the internationally agreed Biosafety Protocol. 1.7.4 Maintenance breeding. Plant breeders are required to maintain the genetic purity of existing parental lines and pre-commercial seed supplies year by year for the varieties developed by them. Thus, the genetic identity and purity of the varieties and hybrids can be preserved against various factors affecting the genetic deterioration, viz., genetic erosion, admixtures, selective influence of pests and diseases, etc. The initial handful of seeds obtained from selected individual plants of a particular variety, for the purposes of purifying and maintaining that variety by the plant breeder who has developed the variety and its further multiplication under his own supervision. To provide breeders' seed constitutes the basis for all further seed production. The quality of the nucleus breeder's seed determines the varietal purity of subsequently multiplied foundation seed, which is further multiplied and provided to farmer as certified seed. Unless the nucleus breeder's seed is of highest purity and quality, the seed multiplied from it cannot be regarded as of satisfactory genetic purity. It is therefore of utmost importance that the nucleus breeder's seed is maintained and produced in such a manner that satisfactory genetic purity, identity and the other good qualities of seed are not compromised. 1.7.5 Seed certification. Seed of an approved variety can only be marketed if it meets strict quality criteria. Seed quality standards are laid down in Indian seed standards and policed by agencies appointed by Government of India. The Indian seed certification system offers an independent assurance of quality to growers. Minimum standards apply for varietal identity, purity and germination capacity. In addition, strict limits apply to seed-borne diseases and the presence of physical impurities such as weed seeds. Seed certification underpins health and purity status of the major arable crops in India. It offers an independent benchmark of quality on which buyers of seed and their customers depend. 1.8 Rationale for sui generis IPR regimes for plant varieties. The origin of distinct form of IPR in the form of PBRs has been necessitated as the patent system is found to be ill-adapted to plant varieties. The rules of the system designed to protect industrial inventions to plant varieties encounter technical difficulties. 1.8.1 Self-replicating inventions. Plant innovation is born in seeds whose very use demands multiplication. These products of innovation are biological products which, at least in the case of self-pollinating plants, can make hundreds of copies of themselves in the natural growth process. While from the standpoint of a producer of plant variety, this is a major cause of concern, the users of the technology and potential copiers are millions of individual farms whose compliance with any protection regime is difficult and expensive to monitor. Moreover, the user of the technology are farmers and farming communities, which in many countries like India involves traditional cultural values that affects food security and the livelihood of the rural poor, thus making the imposition of any control a sensitive political issue. 1.8.2 Invention, inventive step and new plant variety. The inherent genetic diversity within a plant variety and the inevitable changes between generations create problems with the description of the invention. Moreover, for producing new plant varieties, plant breeders rely on traditional breeding methods or standard genetic engineering techniques. These techniques are part of common knowledge, and it is obvious breeders rely on them to search and develop new plant varieties. It can be argued whether application of standard breeding methods constitute an inventive step. Even if these techniques required considerable effort and investment, the product they create may not amount to a patentable invention. In these situations, PBR is more suited, as it specifically protects the end product of the breeding process, that is, the new plant variety in itself. 1.8.3 The criteria of novelty. Patent protection of an industrial invention requires to be new, that is, it should not form part of the state of the art. The novelty criterion of the patent system was difficult to maintain where the natural diversity of a plant variety is insufficiently known to the examiner. Also, the patent application requires that the invention needs to be disclosed in such a way as to allow someone skilled in the art to reproduce. Meeting this requirement is not possible in plant breeding, even if someone has access to the same parents and the same selection strategies. It is impossible to breed the same variety. 1.8.4 Privileges under PBR. The PBR system evolved in various countries in consideration of the rights of their citizens and the potential economic benefits in order to increase national welfare. Since the socio-political systems and the structure of industries differ widely, the optimum contribution to increased welfare may be achieved at different levels of protection. The concerns on food security and traditional systems have led to the establishment of exemptions in several countries. For example, many PBR legislations allow farmers to save, exchange and sell seed in accordance with local custom. Under patents, such actions would constitute infringement. Also, while the patent systems do allow some research on patented inventions, but the form and extent of research allowed is based on case law. And so this is more difficult to assess. However, research use under PBR are more clear-cut process. Provisions of such privilege, where allowed, are generally considered to give holders of PBR certificates weaker protection than the patents. This helps explain why, where the choice is available, commercial breeders often prefer patents or patents plus PBR over PBR alone. 1.9 Justification of PBRs. The primary justification for the establishment of PBRs, like any other form of IPR, is economic. As we have seen above, plant breeding has moved on from being purely an art, i.e., experience and good ends of selection, to become a science-based state-of-art technology enterprise. The private sector involvement expanded with increasing application of practical biotechnological tools for developing new varieties, and multifold increase in private investment resulted in pressures to expand PBRs in agriculture. This point was primarily because these firms aimed to offer agricultural technology as an integrated package, in which improved varieties of planting material were the critical components, offering a type of monopoly for the commercial exploitation of plant variety to the breeder is expected to earn back investment costs and provide an incentive for creating new varieties. The system can also offer additional, more widespread, economic benefits such as reducing transaction costs and clarifying ownership. This is important to provide the farmers and growers increased access to the best available overseas varieties. The issue of PBR is significant and can be understood in two ways. First, in the narrow sense, where protection of plant variety is important to provide an incentive to commercial breeders for developing new plant varieties, and second, in the broader sense, where plant variety protection has direct linkages with the rights of the farmers who have traditionally been breeding plant varieties as per their local conditions, accessibility to plant genetic resources, and concerns related to food security. Farmers have played an important role in the conservation of PGR and will continue to do so in the future as well because of their enormous contributions in the past, present and future towards the conservation of PGR. Farmers have an important stake in any legal regime on plant variety protection, and such a regime should take into account their interests and concerns. 1.10 Summary. Times the Plant Breeders' Rights, PBRs, providing intellectual protection to new plant varieties have a recent origin in the history of IPR accelerated across countries by the acceptance of the TRIPS agreement. Times taxonomists have described, classified, identified, and named plants from early days. While the rank of species are one of the basic units in botanical classification, the plants within a species can be very different. Farmers and growers use a more precisely defined group of plants, selected from within a species, called a plant variety. Times the genetic variability within a species is essential to maintain its reproductive vitality, resistance to disease, and ability to adapt to changing conditions. Systematic selection aimed at selecting the naturally occurring variants formed the basis for earlier crop improvement efforts. Times Mendel elucidated the genetic mechanism involved in inheritance and linked the genotype of plants with resultant expression of the genetic structures as observed in its phenotype. This led to refinement in breeding techniques by crossing carefully chosen parent plants, then selecting the best plants from the resulting offspring to be grown on for further selection. As breeders aim to combine a range of traits such as high yield, quality, and resistance to disease in one plant, breeding of crops with better performance is an extremely lengthy process. Times the speed, accuracy, and scope of the breeding process have been enhanced with the use of recombinant DNA techniques. The direct manipulation of DNA or genetic engineering is regularly being used to complement classical plant breeding. Further, the advancement in science of genomics has broadened the scope and precision in breeding and accelerated the pace of commercial breeding. These techniques allow plant breeders to develop transgenic varieties that have genes inserted into them that can be derived from a wide range of living sources across kingdoms. Times, in addition to plant breeding, commercial seed production entails operations and responsibilities such as source seed production, seed multiplication, quality control, compliance with regulatory requirements, and marketing, etc. The primary justification for the establishment of PBRs, like any other form of IPR, is economic. As plant breeding has moved on to become a science-based state-of-art technology enterprise, the private sector involvement and investment has expanded multifold. The PBRs are expected to earn back investment costs and provide an incentive for creating new varieties that is important to provide the farmers and growers increased access to the best available varieties. Thank you for listening. We will be back with the next chapter.